Okay, so I'm recording now. Okay, so here basically for the markdown file, this is how we specify the document header. Okay, and the output here is going to be whatever. So here, what I'm writing at the script here is to say that I want to download the gapit library. So you can see that the file ends in gapit.library.r. So if you copy this link, okay, and you head over to a browser and you click on a new tab, you open, you see an R script, okay. So basically these lines of code is supposed to install some packages, okay. So those packages are more like R scripts. They are R scripts actually, or dependencies. So here you have gplots, you have LD heatmap, you have genetics as a package, you have ape, you have compiler, grid, big memory to handle memory loss issues, EMREML, you have scatterplot 3D. So these are all packages that Gapit depends on in order to run the analysis for you, okay. And then you also have BioManager. Okay, so these are all packages that Gapit depends on. Now, in my experience, if you try to run this scripts at once, you might have some scripts or some packages that would not install. Okay, and to provide more like a fallback, just that any package refuses to install, I created this install.packages script. Okay, it's just a short script that uses a GitHub repository to install especially the LD heatmap, because from my experience that did not install from CRAN. Okay, so what I did was to add that line of code into the script. So if you see within your markdown file which I provided, you have this source script install.packages, okay, or you can just use this one. This was the latest one I updated, source install.packages.r. So once you run this, it's going to go ahead and then install all of the dependencies that was also available from the online version. But remember, I've done some background work to make sure some of the packages like LD heatmap install. So if you do the online one, which usually you can do with this line of code, so once you click this button over here, it's going to go ahead and then you see that it's installing all of those packages, okay. Or you can also use the offline version, which you can do by running this line of code. So you just come to this line of code and you press Control Enter on your keyboard, okay. If you're on MacBook, it's Command Enter, but if you're on Windows, it's Control Enter, and then it's going to go ahead and install all of those packages for you, okay. Okay, so it's doing all of that work to make life a bit easy, okay. So once it's done installing the package, it's going to take a while, and yeah. Now, if your internet connection is not too strong enough to install the packages, I provided a fallback for you, where I have already, I think I should just allow this to finish running, and okay, maybe I can just stop it. Okay, so that I can continue with the tutorials. Okay, so if your internet connection is not too strong to install all of the packages, I have a fallback to more like have all of the libraries. Okay, so what I did was to install the packages myself and then have them within my global environment and save all of them as an R data file. So all of the functions you find within the R all of the functions that are necessary, so all of the GAPIT functions, the farm CPU functions, and all of that are necessary to run this analysis is stored within this particular file. So it's called GWAS.r data, and it should be inside the same folder as your script. So I'm using this function here to just load that load all of the functions and the data sets that is responsible for performing this. So if you watch over here, I am going to try to remove these, and once I run those that function is going to bring back everything again. Okay, so yes, so basically to remove everything within my global environment, I'm just going to say rm equals list and then ls. Okay, so basically it's going to remove everything within the global environment. So you can see over here environment is empty. So if you've not done anything up to this point, we are on the same page at this point. So I'm not doing any hidden algorithm behind the scenes. Okay, so I'm just trying to say that you can walk along with us if you really want to. Okay, so here all you need to do is to click on this green button. So you might be able to see the green button over here, or you can just press Control Enter or Command Enter to run that line of code. And so once you run that line of code, you see that it's going to fill up the global environment. So all of the data sets, all of the functions or the packages that are necessary to run GWAS with GAPIT is loaded to your global environment. So it makes it easier to work, easier just to follow us. Okay. Okay. Is anybody having any challenges at this point? Okay, good. So here, after going through all of this, after having all of the GAPIT functions and all of the libraries available, the next thing to do is just to run GWAS. It's pretty simple, okay? And so to run GWAS, what we actually need is a function, okay? And that function is a GAPIT function. So here you have GAPIT. Okay, you have GAPIT, and you have like these things are called parameters. Okay, and these are what we saw in the presentation. Okay, so in the presentation we saw that we had a number of models, right? Let me see if I can find that easily. We saw that we had a number of models to actually run GWAS. Yes, so these are they. So here we have the y variable. So the y variable here is the response variable or the phenotype. Okay, so that is the phenotype we have over here. And where is this phenotype coming from? The phenotype is actually a data set. The phenotype is a data set. And where is that data set coming from? The data set is coming from this particular link. Okay, so with your projects, or if you are running your own analysis, you would have the data set stored on your computer. So this is the data set. Okay, so this is taxa, or a sort of classification for individuals. So you have ear height, you have dipole, and you have ear parameter. Ear diameter. Okay, so here these are going to be your phenotypes. Okay, and then here you have a GD data set, which you would also have access to once you do your genetic data analysis. So basically it's like a no, this is This is a genetic data set. So you have the 2s and the 0s and the 1s. So basically it's a genetic data set. The AA, AT, GC we saw usually are coded into 1s and 0s. And then we can now use those 1s and 0s to perform our analysis, okay? And you don't do that manually. Usually you have a script or a package to do that for you. And then over here as well, we have another file, which is like a relationship matrix, okay? So you have another file that contains, is more like a genotype file or an allele file. So what it does is that it contains your SNP, the SNP you are interested in, the chromosome it is located on, and then the position it is located on that chromosome. Okay, and so this file is usually mapped to this particular file. So if you look very here, you have PZB 00859.1, and then you also have the same thing over here, PZB 00859.1. So we are mapping this SNP, the chromosome and the position, to these data sets which contains the genotype, and that is how we are able to have the phenotype and also the genotype. And then we also have a third file, which is the HapMap file, which most of the times will be your VCF file, which have been converted to HapMap. And here, this is where you have your AA, your CC, your TT, and whatever be the case. And sometimes you might have NN. Usually it stands for missing data, okay? And yeah, basically that is it. So if you run these codes individually, or you go to the files tab, if you are able to download the data and you go to the files tab, you see that these files are also there. So you can see ndp trace.txt, you can see mdp numeric.txt, you can see mdp snp information.txt, and they are also contained within the data file, okay? And so once you have those file sets, then the next thing to do is to run a simulation. So GAPIT requires you to run a simulation of the phenotype, okay? Put all the genotype information together and the heritability of the traits. So you can see H2 over here is equal to 0.7. So before you perform these tests, usually, before you run GWAS, usually you should know the heritability of the trait. And you do that by computing heritability with the genotype, okay? The genotype and the phenotype. And then you get a SNP-based heritability, and then you can impute that value over here. And then here, this value here is called the QTNs, quantitative trait nucleotides. So basically it's the SNPs, the number of SNPs you are looking at across the genome. And then here you are looking at the genotype file, and then one of them is a genotype file, and then the other one is the SNP file. And then you run that into this variable called my simulation, and then you extract the y variable from my simulation into the my y variable, okay? So I'm going to run this one after the other. So I run this. Set seed basically is to like create is to save that particular this particular process, so that when I run it again I don't get a different result, okay? So I run my simulation, and then it's going to use, it says it's using MLM to simulate that data, okay? And then once it's done, you see that I have a variable in an object in my global environment here called my simulation, and then you have all of these data sets from that, okay? And then from there we extract the response variable, okay? So you can see the response variable over here, the taxa, and all of the different traits that are coming from there. So we pass that to y, okay? And then from there we can use all of those to perform our GWAS analysis, okay? So here I'm just going to go ahead and click run on this. Okay, so after clicking run, it's telling me that it can't find a function called plotly. Okay, so I think I did. Let me check on that. Okay. Yes, so I actually provided a backup for that. So here, all we need to do is to run this code. I'm going to copy it and bring it over to the GWAS.rmd file and then run that again. So here, I'm going to run this code again, Control Enter or Command Enter to download Plotly, to install Plotly, and then after we are done installing Plotly, then I can go ahead and then use the library file to the library function to load Plotly in. So basically, it's to plot all of the outputs from GWAS. Okay, that's what Plotly is going to do. So to plot all of the output from GWAS so that you have all of those graphs in the PDFs as you would see in a minute. Okay. So here I'll go ahead and then run the GWAS. And if everything goes right, we'll be having all of those files created in a minute. So you see that it's calculating kinship, it's creating ngtree.pdf, whatever. So that's the power of Gapet. It does like all of these things for you. You don't have to like calculate kinship manually. You don't have to estimate blues and blabs and all of that. And yeah, you can get your results quickly. So how fast this is going to run, it depends on so many factors. So one of them is your computing power. So how fast your computer is, the size of your phenotype. If you have a lot of traits, it's going to take a while to do the associations, and then also the size of your genotype file. Okay, so those are the things that influences how fast it might run. Okay.
Good evening, Doc.
Yeah, good evening, sir.
Please, with the package installation, I
Okay.
think there was a command that helps us to run the entire package that's needed for this particular analysis.
Yeah.
I
Yeah.
wanted.
Yeah. So that's this line of code. Source install the packages. Install packages.r.
Okay.
Yeah.
Okay, so when I tried to run that, there is an error that is saying that the installation of package in quotes later had non-zero exit status. So there is also a warning that this package has a configure script and it probably needs manual configuration.
Okay. Can you share your screen with me?
Okay, sure. This is it.
Oh, okay. Let me see. All install the packages. Blah blah blah. Okay, so what is actually going on is that you need a newer R version to run that particular package. So it's having a package conflict.
Oh, okay.
Yeah. Yeah. So you might just want to uninstall your current R package and then download and install a new one. Your base R, okay? So that's what's going on.
All right, thank you.
Okay, so GWAS is done running, and let me just share the screen. GWAS is done running, and we can actually see the results of the GWAS file. Okay, yeah, so we have that here now. So we can see the results of the GWAS file from here. So now these results will be set to whatever you have as your working directory in R, okay? And so sometimes some people have their working directories as their download file, their download folder or document folder and so on. And so once they are done running, they are not able to see the files, and they are sort of like confused as to what is happening. So what you want to do is to make sure that you set your working directory to be your current directory. And to do that easily, you can come over here to where you have more, and then once you are within your GAPIT folder, and that is where you want to run it, you can just click on set as working directory. Then you copy the code, and then you bring that code over here. Okay, now mine is using /, okay, but if you set your working directory as I've shown you over here, if you're on a Windows PC, it probably is going to use /. Okay, and that's what I'm showing you how to do it. So once you are done, once you set it, then you can just copy whatever code you have over here, and then you bring it here, so that in future you're going to know that when I run GWAS maybe on the 24th of February, I run it from this particular directory, and then you can go there to look for your results or your files. Okay, basically that is what is happening. Okay, so here we have like all of these results, or all of these PDFs, as you can see, from the GWAS analysis. Okay, and yeah, you are going to have all of that. And what is happening is that the reason we have so many files is that GAPIT is using like a lot of models to run GWAS. So we remember we talked about the generalized linear model and the mixed linear model. Okay, aside the generalized and the mixed linear model, GAPIT is using SUPER, which is also another model. Is using MLMM, is using FAM CPU, which is like one of the most powerful models we have like today for performing GWAS, and then it's also using Blink. Okay, and then it's going to use all of the models to fit GWAS, and then show which is the best one for fitting GWAS based on your phenotype, your genotype, population structure, and kinship matrix. Okay, so basically that is what is going on over here. Okay, so here I'm just going to move back to the presentation so that we talk about some of the results we have from the GWAS analysis, which you should have access to after all of these. Okay, so some of the results you would find, just give me a moment. So these, we've already talked about how the GWAS script. So this is how the GWAS output is going to look like once you are done. So you can get that using this function. Okay, so let me just maybe I can go back up here. So let's see if we can get this. Yeah, so this is it. So the view function basically is to view whatever object, but most of the times if you use view on a vector, it's not going to work. So it has to be like a data frame. So if you use the view function on a data frame, you are going to get the result. So that is how the GAPIT outputs looks like. Okay, but you can also use the view function to get it. So if you are using the view function and it's not a data frame, you can just use the class function to check if that particular object you are running is a data frame or is a list. Okay, here it's a list, and so because it's a list, if you run it, you are going to see like the output of the GWAS. Okay, so this is like one of the main outputs. So you have the SNPs, you have the chromosome number. So here we are on chromosome 1, the position of the chromosome, the p-value for that SNP effect, the minor allele frequency for this particular SNP. So that's the ratio of this SNP to the rest of the SNPs. Okay, and then you also have the effect sizes. Okay, so basically that's what like one of the outcomes, or the outcome that GAPIT is using to create all of these other files for you. Okay, so that is what you have over here in your short. Okay, and over here you have the heritability of the trait. So GAPIT also more like computes a heritability value. So you have the genetic value, which is based on the SNPs. So sometimes people call this narrow sense heritability or SNP-based heritability. So if you add the genetic value to the residual, which sometimes people refer to as error, and you divide the genetic value by the residual, then by the total, then you would get the heritability of the traits. Okay, you get the heritability of the traits. Okay, so here let me clear this. So we have 178.11 plus 126.84. okay, so that's 304.95, right? And then we divide 178.11 by 304.95, and then you see what the heritability of that particular trait is. Ear height, okay, is 0.5. okay, that's the narrow sense heritability of that trait. Okay, and then what you have here is a heat map showing the kinship matrix. Okay, so if I zoom into this particular image, I don't know if it's going to go like way more than this, but if I zoom in like really well, you will see that what you have over here are the SNPs, or the individuals. Okay, because kinship is basically based on individuals. Let me see if I can find the kinship matrix from the PDF file and show that to you. Okay. .genotype.kin. Yeah, so that is what you have here. Okay, here it's not showing us PDF. Let's go to okay. Okay, so here you can see it's from the PDF file that, yes, so zooming in really well because there's a lot of individuals, so it's kind of like really small. So here you can see these values here represent individuals. So it might be plants, it might be bacterial types, it might be whatever. Okay, so basically a relationship between individuals, how those individuals relate together is the kinship matrix. Okay, and this is just the heat map of the kinship matrix. And then you have some other plots that would usually come out from GWAS. Let me zoom out a bit. You have some other plots that will usually come out from GWAS. So this is what we call PCA, or population structure. So if you watch carefully, you have some individuals or some PCs, some values clustering around this point. You have some clustering around this point, and you have some clustering around this point. Now, this sort of cluster, once you see the cluster, it means that there's some sort of population structure that went on in time past, like between the individuals. It means that some individuals were moved to other environments and they became adapted to those environments, and so their allele frequencies have changed over time, and that is what we can infer from the population structure. Okay, and so it's important to add, once you see something like this, it's important to capture your eigen vectors and then add them to your GWAS analysis, your model. Okay, and then you have the GWAS results in a table, which is not so interesting most of the time. So we want to have it in a Manhattan plot, and you should also be able to find that results within your directory. So you can see things like GWAS over here. So these are CSV files. Let me look for the PDF files. Yes, so you can see something like this. This GWAS was computed. This GWAS was computed based on the Blink model, and there are so many other models. Depending on what you want, you can use one of those. Okay, so here you can see that we have an association, okay, with the traits. We have this particular SNP occurring over here that has a very high p-value, and it tells us that that particular SNP is highly associated with the traits. Okay, or at least these two or three SNPs are highly associated with the traits. Okay, and so that's what we would usually get with a GWAS analysis. And then when you are done, you can decide to save whatever plot you create, or if you use Gapit will save them as PDFs, and then you can use whatever tools you want to use in extracting the plots and then using them for your publication. So basically that is it for the practical part of it. Yeah. So once again, if you have any questions, I'm open to your questions. So I'm currently recording the session. Once I'm done, I would render it and then share as an so I'll upload to YouTube, but not a public YouTube. So more like an unlinked YouTube, which people cannot easily get access to, and then share with Dr. Kwanzaa, and then she can share with you guys. Okay, so are there any other questions? It might feel like a lot, I know. I mean, when I started learning GWAS for the first time, it was a lot of concepts to like grasp, and you might want to go through each of the concepts like one after the other, understanding the models, understanding kinship, understanding PCA, and all of that. And once all of that knowledge comes together, understanding GWAS, it's a possibility. Like, it becomes really easy to do so. Yeah.
So, Mr. Caleb, what I want you to probably explain. So, as MPhil students, if they want to do a topic in GWAS, can you give examples of what they can do?
Yes. So, a topic in GWAS. Okay. So, can you give me an Okay, so a topic in GWAS usually depends on the area. So I'm coming from plant breeding and genetics, and so I'm usually looking at plants and plant traits. But some other people coming from medical genetics might be looking at looking for variants in diseases. Okay, so with biotechnology, I'm not too sure the application of biotechnology they are looking at. But if I get to know the application of biotechnology, then I can suggest like what they might be looking out
for. Very diverse. They can apply it in plants, in microorganisms, in medicine, in technology, almost anything you can think about.
Yeah, once you have a genotype and you have a phenotype, you can apply GWAS. Yeah.
Okay.
Yeah.
Please ask the questions, so dear students. I'm not the one to ask the questions for you. Okay, since they are not asking, I'll ask. So does it mean that for GWAS, you have to always do genome-wide sequencing? I mean, for any organism that you are looking at. Is that it?
Yeah, so yes and no. You if you don't have access to the genome, usually that will be your first. Usually, what people do is to look for the genome. Let me just share this resource with you guys. NCBI. Yes, so this is NCBI, and most people usually would come to NCBI depending on what organism they are working with, and then search through NCBI for a genome. Okay, now once they find a genome, then the individuals they are using, they can map the individuals they are using to the genome they have found. Then they can use the genome from NCBI to actually perform GWAS. But if you don't have access to that, then you can either work with a collaborator, or if your funding agency or your grant agency has like enough money to do genome sequencing, then you can do genome sequencing either using GBS, genotyping by sequencing, which is relatively cheaper than the whole genome sequencing.
Okay.
Yeah.
So what it means is that they can use already sequenced genome.
Yes.
But however, they still need to carry out some wet experiments. Is that it?
So if you have access to already sequenced genome, you don't need a wet experiment. most of the times you don't need a wet experiment. So the wet experiment they might need is basically to phenotype, but not a wet experiment to extract DNA, do PCR, and then go ahead
So
to
when I did wet experiment, I wasn't thinking of DNA.
genotype.
So
Okay,
for
okay,
instance, for instance, you want to look at, there are different let's say millets. There are
okay, okay.
different, I don't know whether to call it ecotype or
Ecotypes,
land
yeah,
races.
botanical types, yeah, yeah.
So maybe someone would really want to look at, let's say, is it environment that makes them to be different? So
Okay,
in this case, does it mean that they will not grow them to probably try to phenotype to see which one flowers and all that? So that's what I mean by wet
wet
experiment.
experiment. I understand you now. I understand you now.
So my question is that, does it mean that one doesn't even need to do that? All that they need to do is to go into NCBI and then look at whether all the different ecotypes and northern region has been sequenced, and then they can work with it. Is that it?
So if you are GWAS needs genotype and phenotype. If you don't have access to the phenotype data, you definitely need to do like your wet experiment. So you need to plant the different ecotypes of the millets, and you need to now map those individuals to an already sequenced genome. The challenge really is that, especially for our part of the world, we are not able to have like one-to-one maps of the genome of the individuals we are looking at, and those individuals contained within the genome. So usually for people who work with this sort of analysis, they use certain individuals, right? They use genotypes. I don't want to use the word genotypes because it confuses with the allele genotype, but usually they use plants or individuals to sort of like do this analysis and then extract their DNA and then genotype them. Okay. So our problem usually is that our varieties are not more like standardized to be able to map them to these very same genotypes that are already on NCBI. But if you do a bit of research and you are able to find a map, so maybe if, for instance, in maize in Ghana we have abontem, right? So if you are able to find a map of what abontem is to an international reference, then you can easily find the genome, or it becomes easier to find the genome of abontem and then use that for your GWAS. Sort of. That's what I mean.
Okay.
Yeah. So that's one cheap way to actually go about some of these. Yeah. But you still have to do your planting, and then you still have to extract your data. And then there's also phytosome. There's also phytosome where you can download genome files from. Someone has a question. Akume, please go ahead.
Yeah, thank you. I wanted to find out, is there any limitations of GWAS in detecting rare variants?
Yes, so from my presentation, I mentioned some of these, not accounting for population structure. It's one of the limitations we've found so far. Not accounting for kinship is one of the limitations. Another limitation, using the wrong model might also impose a limitation, and that's why with GAPIT you run a lot of models to see which one can have like enough power to detect the SNP, okay? And then also not phenotyping accurately. If you don't have an accurate phenotyping, it might also pose as a limitation, and then not genotyping accurately. Yeah. So these are some of, and sometimes you might do everything right and still things goes wrong because you are looking at the wrong traits. When I say wrong traits, I mean that the variants that are responsible for those traits might not be SNPs. They might be a large insertion, they might be an inversion, they might be like with maize, we have a lot of this. We have this concept of transposons with maize, where genes move from one region of the genome to another region of the genome. So you might have some transposons that might limit your ability to find SNPs, and also the genetic architecture of the trait. So before you get to the GWAS point, you should have studied all of these, and then usually you should capture them in your paper, and that is what some of the things the editors are looking at when you write a paper for GWAS. You're looking at the heritability of the trait. You're looking at whether those traits, the genetic architecture of the trait, whether it's additive, dominant, or epistatic. Usually it should be additive if GWAS is going to work. You're looking at the type of model you want to use to detect that trait. And then you're looking at your kinship structure, your kinship relationship, genetic relationship matrix or kinship. You're also looking at whether the population do have population structure. You show that by doing PCA, and then you add your PCA, the result of your PCA computation into your model. Now, we are not doing all of those things manually because GAPIT does all of those things behind the scene. We just have to provide the files to it. But if you are using some other softwares like LDAC or Plink, you might have to run each of these steps manually and then get the results and then use that for your GWAS. Yeah. Okay, thank you, John. Yeah, any other questions?
Hello, sir.
Yeah, hello, sir.
Sir, thank you so much for this wonderful presentation. Sir, I would like to know how could we use GWAS to address the occurrence of false positives?
So when you say false positives, what do you mean by false positives?
All right. So one, like in terms of relatedness.
Can you say that again?
Come again, sir.
Can you say that again? I did not get the last word.
Yes. So what I'm trying to say is that how do we use GWAS to address the occurrence of ancestry false positives? I've read something like ancestry, when you mentioned about the confounding and all of that stuff. So GWAS, how can we use GWAS to address? I think it's a concern, false positives and relatedness or heritability.
Okay.
I don't know, am I making sense?
Yeah, I get what you mean now. So usually,
okay.
Yeah, false positives in terms of relatedness or co-ancestry or kinship is addressed using the kinship matrix. So once you compute your kinship matrix, the model gets to know how each individuals are related to each other, and those values usually is the coefficient of inbreeding. So it relates each values based on their ancestry and computes the coefficient of inbreeding for each of them, and then you have a kinship matrix that has all of the coefficients of inbreeding, and then you use that, you add that to your GWAS model when you are running the model. Now, some other false positives might arise depending on what you use for your threshold. So either using the p-value or using the FDR. So the FDR is more like a stricter measure. It's called the false discovery rate. And then some people also use the Bonferroni correction factor to control for false positives. And sometimes you might do all of that right and still not be able to find the exact SNP. But what GWAS does is to give you a lead. Okay, so it's just like a criminal investigator. Once you do like a DNA and all of that, it gives you a lead as to what is going on, and then you can now narrow down your search as to where those genes are located or which individuals have those genes. And so you go ahead to do techniques like CRISPR, which I mean for now I know is expensive, but if you have collaborators working in other countries, in advanced countries, you can have access to those. And then also there are other ways actually to go around that, using biparental crosses to map the gene, fine mapping, or using nested association mapping. So these are all techniques to sort of go ahead to confirm if the SNP you've found is actually the right SNP or not.
All right, thank you very much, sir. Please, sir, I would like to refer you back to the Manhattan plot you mentioned about. Like, what is it all about? I think I'm lost there.
Yeah, so the Manhattan plot actually shows, so the values or the dots in colors over here are all SNPs. So they are alleles or variants, okay? So all of these dots you are seeing over here are SNPs. Now the y-axis, is the negative log 10 p-value. Now the reason we use the negative log 10 p-value is that the p-values themselves are really large. When you run GWAS, if you look at the p-value, let me see if I can show you like a p-value for GWAS. So you can see the p-value for GWAS, right? 0.00024, 0.00, and sometimes it can even be more. And this does not mean that those SNPs are significant. It does not necessarily mean so. Okay, so what we do is to take the negative log 10 of those p-values, okay, so that we can plot all of the SNPs that we actually find, okay, on this particular plot. And what you see on the x-axis is the various chromosome numbers. So this is chromosome 1, so these are the SNPs on chromosome 1, chromosome 2, chromosome 3, all the way up to chromosome 10. Okay, all the way up to chromosome 10. So you have all of those. So if you find a value or a SNP with a significant p-value or with a very high p-value, the x-axis helps you to know which chromosome that SNP is located on. Okay, so if it's located on chromosome 7, you know. If it's located on chromosome 1, you know that it is located on chromosome 1. So basically, it's a relationship between the negative log 10 p-value and the chromosome. And what you have on the Cartesian is basically your SNPs, the various SNPs you have within the genome. Yeah.
All right, thank you, sir.
Mm-hmm. You're welcome.
Hello.
Yeah, hello.
Sorry. So, I think Dr. Lydia once gave us some assignment, and then when I had a research article or a paper, so I saw that this Manhattan plot was there in that research, and also I saw farm CPU. So, and I think when you run the test, I saw some of those farm CPU files. Maybe if you can pick one of those data to explain that.
Okay, sure. So let me see if I can find the one with from CPU. So this particular plot has all of the models, okay, and their Manhattan plot. So you can see that was what I was referring to. So this is from CPU, right? And then this is Blink, okay? So usually it starts with a base model or the null model, which is a GLM, okay? And then it looks like it's so Gapit has used all of these models to run GWAS, and then it's telling you that, okay, this particular model is able to detect these SNPs, or this particular model is not able to detect these SNPs. So here you can see we have the GLM, and here the model is what? The model is GLM, right? GLM.sim. Sim just means simulation, Okay, and then here we have MLM, which is the mixed linear model. So this is a generalized linear model. This is a mixed linear model. Then we also have another model called super. Okay, and then here we have another model called multivariate linear mixed model, which is also another form of model. And then we also have from CPU, okay, and then we also have Blink, okay. And then you can see that to some extent, some of them are able to find certain variants which the others cannot find. So you can see that from CPU, it's able to find these dots. It's saying that these dots appears above the threshold, okay. And then if you go to the other, so if you go to the other chromosomes, some of them might not be able to find them, okay. So some have like better algorithms or better statistical power to detect the presence of SNPs or the presence of variants than others will do, okay. So sometimes people just like with GAPIT run everything and then find which one is better or is a better fit than the other one, and they are actually objective values to check for those. That's the AIC, the AICC, and the BIC to check, and usually the lower value the better. Yeah, so basically that's what it's happening. It's just a model use or it's just what's it called, an algorithm using computing GWAS. Okay, you're welcome. Okay, so if there are no other questions, I would like to end the session here and say thank you all for coming, and I hope to see some of your publications in the future featuring GWAS and pushing the boundaries of science. Thank you all.
Thank you so much.
Mr. Caleb. We are grateful, and we hope that the students can network and we'll see how this goes. Thank you so much, and have a good day. I'm sure you are still in your afternoon. So
yeah, it's not yet 2:00 yet.
Yes. Okay. Thank you. Thank you. We are grateful.
Thank you so much. Bye, everyone.
Okay. Bye for now.
Mm-hmm.